welcome to a very, very special moment. My name is John Gallin, and I'm the director of the NIH Clinical Center, and I have this distinct pleasure of welcoming all of you to the beginning of our 60th anniversary celebration. Indeed, on July 6, 1953, the first patient was admitted to the NIH. And we have a very special program today. I'm going to try to give you a little bit of a snapshot, telling you about our past. And then we have what I believe is the very first example of a father-son pair of speakers at Grand Rounds in the history of the NIH with Jim and Steve Holland. And I'll tell you more about that in a moment. I also want to take a moment now to just thank the Foundation for the Advanced Education in the Sciences, or FAES, who after this event are providing a little bit of refreshments in the South Lobby, and you're all invited. I told the 88 new fellows who came yesterday for their arrival at the NIH not to miss the opportunity of a free lunch, because it doesn't happen often. So as I began to think about what I might say on this day, I began to think, well, what does NIH mean to different people? And it really means different things. To some, it's the buildings and the grounds. To many, it's the science. To the patients, it's the care. But I think to everyone, it's the people, the amazing group of people who work here, who we get to interact with every day and who have been here before us. Yesterday, when I did the introduction to the fellows, I showed a few movie clips, which I decided to show you, setting the tone for when the Clinical Center opened back in 1953 and the years just before that. So if I could get our projectionists to start this presentation, we would like to show a speech that Franklin Roosevelt gave here in 1940.
The President of the United States dedicates the National Institute of Health in Bethesda, Maryland. 6 hours ago, the presidential special train rolled into Washington's Union Station, bringing the president back from his one-day New England tour, which ended in Boston last night. And now the president has come by automobile to the National Health Institute in Bethesda, Maryland, on the outskirts of Washington. Here, on a pleasant Indian summer afternoon, a moderate-sized audience has gathered before the white-columned, red-brick Institute Administration building, set in the green rolling Maryland hills and surrounded by trees thick with red and yellow autumn leaves.
Ladies and gentlemen, the President of the United States.
Mr. Coy, Dr. Thompson, the Governor of Maryland, Governor O'Connor, ladies and gentlemen, nowhere in the world except in the Americas is it possible for any nation to devote a great sector of its effort to life conservation rather than life destruction. All of us are grateful that we in the United States can still turn our thoughts and our attention to those institutions of our country that symbolize peace, institutions whose purpose it is to save life and not to destroy it. It is for the dedication of these noble buildings to the service of man that we are assembled here today. The National Institute of Health speaks the universal language of humanitarianism. It has been devoted throughout its long and distinguished history to furthering the health of all mankind, in which service it has recognized no limitations imposed by international boundaries and has recognized no distinctions of race or creed or color. The total defense that we have heard so much about of late That total defense which this nation seeks involves a great deal more than building airplanes and ships and guns and bombs. For we cannot be a strong nation unless we are a healthy nation.
Okay, thank you.
So we must...
This clip is available, and the Library of Medicine kindly lent it to us for today, and I'm sure if you are interested in seeing it, we can make that available in toto. But I think it emphasizes back in 1940, before the clinical center had become anything, not even a twinkle in an eye, people were talking about the importance of the health of the nation. And the NIH Clinical Center was first authorized in July 1 of 1944 in the public health law at the time. It was appropriated 3 years later in 1947, and the cornerstone for the new building was laid by Harry Truman in 1951. And when Truman came here, if we could have the clip, the next movie clip, I guess I could do it.
This clinical research center will advance the work that is being done by all of us to achieve better health. Chronic illnesses take a tremendous human toll. Modern medicine must find ways of detecting these diseases in their early stages and of stopping their destructive force. That'll be the major work of this clinical research center.
He then also went on to say, the greatest groups of scientists ever gathered for basic and applied medical research to serve for many generations as a monument to our desire for human health and happiness in a world at peace will be here. And how right he was. So I show this picture of what the clinical center looked like right after it opened. And I told all the fellows we play golf. This center, as you can see, was in the center of a golf course, which sadly had to be removed with progress. So opening day, as I mentioned earlier, was on July 6, 1953, and the very first patient admitted on that day was Charles Meredith, a Maryland farmer with prostate cancer, along with 17 other patients who had the diseases of cancer, heart disease, and arthritis. The cost of the new clinical center was $850 million, or $64 million, or $850 million in today's dollars. So it was actually more expensive than the Marco Hatfield Clinical Research Center, which was about $600 million. But of course it was a little bigger. I point out the last line on this slide that the opening was actually delayed for a year because the Budget Bureau's refusal to provide operating funds, because I'm told there was concern about the operating costs of this clinical center. So it doesn't sound like times have changed very much over the 60 years. This is a chart depicting what went to President Truman in 1954 as the president's budget, showing the bed distribution and nursing units for the different institutes at that time who were here: cancer, mental health, heart, arthritis, micro, and neurology. And you can see that the largest number of contingent of beds, 35, was to the cancer, second to mental and heart and arthritis and micro, and last to neurology. So 150 beds on opening day at 8 nursing units. And as of a year later, it was to expand to 300 as distributed here. So the legacy of distribution of beds, which was so important in the first, about 50 years of the clinical center, are shown here, where they were assigned patient care units to each institute. That, of course, changed when we moved into the Marco Hatfield Clinical Research Center, where we instituted a new policy called sharing. and that had its own challenges. So lots have happened. As a matter of fact, as I was thinking about what to say, and I asked all the clinical directors to give me a list of their accomplishments, the list was so enormous and so large that I rapidly had to give up trying to display it here. So I've just listed a few to overwhelm you, to give you a sense of what happened in the first 50 years, and then I'll show you a little bit what happened in the last decade. But they included, at the very first meeting of the medical board in 1953, a recognition of a need for formal review of clinical protocols. And those minutes, which are available if anybody wants to see them, articulated informed consent and the whole review process, and later were adopted by the Surgeon General of the United States to become the IRB process for the United States. Also with the opening of the clinical center very early was the creation of a normal volunteer program, which so many of us in this room have taken advantage of in terms of enriching the clinical research opportunities. The specific examples of accomplishment are lumped in some of the following bullets. Chemotherapy for cancer, first for choriocarcinoma, then childhood leukemia, and Hodgkin's disease. The use of blood lipid levels of biomarkers for cardiovascular disease. The use of leptin to treat lipodystrophy. The use of nitroglycerin for heart attacks. The use of enzyme replacement therapy for Gaucher's disease, later leading to the founding of Genzyme Corporation. The first artificial mitral heart valve. The use of immunosuppressive therapy for non-malignant diseases. First use of fluoride gels to treat dental caries as an infectious disease. The first electronic medical information system, which we called MIS. The first treatments for AIDS, the first 3 drugs. The first blood tests for AIDS and hepatitis, leading to providing a safe blood supply for the United States. The use of acyclovir for genital herpes. First gene therapy for adenosine deaminase deficiency. The use of lithium for bipolar disorders. The diagnosis and treatment of pediatric obsessive compulsive disorder, childhood onset schizophrenia, severe mood dysregulation, and the pediatric autoimmune neuropsychiatric disorder associated with streptococcal infection of pandas. All these started here. I mean, so much of the standard of care in the United States, which we take for granted, originated in this building. So why did everybody succeed? I thought about that, and last fall, Lynn Loriot, who was a former clinical leader in the Child Health Institute, had a slide that I've modified a little bit. First of all, there were lots of smart and creative people. And the investigators who were here literally had the pick of the litter of the young investigators from medical schools when it came to their trainees and fellows. They had secure salaries, and failure was an acknowledged part of the scientific process. So you didn't worry that if your studies failed, you'd lose a grant. Investigators were in the lab and on the ward every day. And it was a great patient care environment. That fourth bullet, I think, may be the most significant one, and one that we always struggle to try to preserve and enrich. And research questions were guided by the human condition, experiments of nature, the rare diseases, bedside to bench and back. So what happened in the last decade? Well, here's a list of some of the milestones of the non-scientific accomplishments. We moved into our new Marco Hatfield Clinical Research Center. We opened the Edmund J. Safra Family Lodge. We brought in a modern informatics system, CRIS, Clinical Research Information System. We built a new biomedical translational research information system to merge the basic and the clinical science together. We opened a PACS system for all our imaging pictures. We computerized the intensive care unit, and any of you who've worked on that unit and saw those huge spreadsheets, all of which were done by hand, they're gone now, and Henry Mazur and his team created an electronic system. We have a new data transformation initiative completed, which gives us the ability to track data with great sophistication. We've got all sorts of wonderful training programs, including a summer camp for PhD students, which is underway right now, introducing them to clinical research, as well as a sabbatical program in clinical research management. We initiated Project Search for handicapped young people to come here and become interns, children with autism and Down syndrome, and about 75 to 80% of those have now become employees on our campus. Our nursing department introduced the whole new field, basically, of international clinical research nursing through Claire Hastings. We've opened our doors to the extramural community, and the first grants for this are under review today. And today, we opened the patient portal, so patients have access to their own electronic medical record. And in recognition of all the wonderful things that you all have done, in 2011, we won the Laska Bloomberg Public Health Service Award. Clearly a wonderful recognition. Scientifically, let me just show you a few things that have happened in the last decade. Immunosuppressive therapy for aplastic anemia was developed by Neil Young and his team and is now standard of care for this disease. Dan Kastner discovered the autoinflammatory diseases. The Deafness Institute identified genes for stuttering. Cancer Institute, the cell therapy for metastatic melanoma, which has become a model for cell therapy for cancer. The first immunotoxin to treat malignancy, hairy cell leukemia. The identification of the genetic basis of kidney cancers with novel therapeutics. New approaches for the diagnosis and management of prostate cancer, and the application of microbial whole genome sequencing in hospital epidemiology. These are all a few of the things that have happened this year. So you all should be proud of this. And of course, it wouldn't happen without our patients. And our patients have been our partners for over 60 years, and they're really amazing. Last week, Francis Collins shared with me a letter that one of our grateful patients sent to him about his experience at the NIH, coming from a small Midwestern city. And I wanted to read these words to you, extracted from a very long letter. I feel that calling this experience humbling, life-changing, or inspirational would somewhat cheapen or not define the meeting. What happened to me at NIH is life. Everyone should experience life in such a place. This patient is still being followed here, and I thought his words captured so much of what our patients feel about this place, this environment that all of you create. So I wanted to give you this brief introduction to highlight a few of the things that have gone on around here, and just to remind you about what's going on at the Clinical Center now and what's happened in the past. And at this point, I'm going to introduce the important speakers today, Dr. James and Dr. Stephen Holland. And let me start with Dr. James Holland. James F. Holland is a medical oncologist and a distinguished professor of neoplastic diseases in the Department of Medicine and Tisch Cancer Institute at the Icahn School of Medicine at Mount Sinai in New York City. He attended Princeton University and graduated from Columbia University College of Physicians and Surgeons in 1947. He completed a residency in internal medicine at Columbia in 1949, and after a tour in the Army, he completed his fellowship in oncology at the Francis Delafield Hospital in 1953. He arrived on opening day of the Clinical Center in July 1953 and worked in the NCI until November 1954 before joining Roswell Park Cancer Institute, where he ultimately became Chief of Medicine and Director of the Cancer Research Center before becoming Director at the Gerald H. Rutenberg Cancer Center at Mount Sinai in 1973. From 1963 to 1981, Dr. Holland served as chairman of the Acute Leukemia Group B later named Cancer and Leukemia Group B, an international cooperative group of physicians and scientists who developed multimodality treatment clinical trials in oncology. Under Dr. Holland's outstanding leadership, the group's investigations resulted in cures for children with acute lymphocytic leukemia, the introduction of cisplatin as a therapy for testicular cancer, and of doxorubicin for osteosarcoma. In 1972, Dr. Holland was awarded the Albert Lasker Medical Research Award for his outstanding contributions to the concept and application of combination therapy in the treatment of acute leukemia in children. In 1997, he was awarded an honorary Doctor of Science degree by the State University of New York at SUNY in Buffalo for his scientific contributions. He is currently an editor of the Holland Fry Cancer Medicine, the first text in medical oncology, which is now in its 8th edition. He's a member of the Expert Advisory Panel of Cancer for the World Health Organization and a co-founder of the African Organization of Research and Training in Africa, known as AORTIC. Dr. Holland's many honors and awards include the Gold Medal of Distinguished Achievements in Medicine from the Alumni Association of the College of Physicians and Surgeons at Columbia in 1963, the National Cancer Society, the American Cancer Society National Award in 1981, the Distinguished Scientific Award of the American Society of Clinical Oncology in 93, and the T.J. Martel Foundation Lifetime Scientific Achievement Award in 2008. He's also elected to the inaugural class of the Fellows of the American Association of Cancer Research Academy in April of 2013, based on his outstanding scientific achievements in cancer research. Over a span of many decades, using molecular methods, Dr. Holland and his team have recently discovered and investigated a human mammary tumor virus in breast cancer. They are studying its possible etiologic role for breast cancer, as well as its potential therapeutic and preventive implications. And today he's going to present the human mammary tumor virus. And now how lucky we are to have one of our first people at the Clinical Center present his current work.
Thank you, Dr. Gallin. It sounded a little like a premature obituary, and I was very, very unhappy to see there's a level place there for an autopsy. I did arrive on opening day of the Clinical Center, and the activity of tremendous interest was lysergic acid diethylamide, which had been used at the Navy in some volunteer sailors. The word volunteer is open to question. And it was of great importance because it was a treatment that was done at microgram levels. B12 had just been recognized, and it was active at microgram levels, whereas other things previously had been used at gram and milligram levels. So these were impressive activities. The paintings made by sailors were equivalent to sailor's art and gave us considerable interest. Put in context, this was 3 months after Watson and Crick had defined the structure of DNA. And before there was any information about message or downstream protein synthesis, and retrospectively, you could say we were pretty primitive. On the other hand, when I got here, I was interested in leukemia, had been interested as a medical student, and could I have the first slide, please? Was introduced to Lloyd Law, who was a senior scientist here, who showed in a wonderful set of experiments, seen on the left, that leukemia with survival on the ordinate, in the yellow in DBA2 mice and in hybrid mice in red, the survival of this leukemia, which he had induced with diethylstilbestrol earlier on, leukemia L1210, was very short. And when he gave two drugs that were known to be active in animal leukemia, methotrexate, the successor name for amethopterin, which was the successor compound to aminopterin, the first drug that was useful in childhood leukemia, and gave it first and then azaguanine afterwards, there was indeed survival extension because of the combination of these two drugs. On the other hand, when he gave them in combination, you can see on the right a significant increase in survival, particularly in the hybrid mice, and that was the beginning of combination chemotherapy. I put that together using methotrexate and 6-mercaptopurine under Lloyd Law's guidance in patients, and we started that trial here at the NIH. About a month before I left the National Institutes of Health, where I had been very happy at $7,200 a year and got an offer of $11,400 a year at Roswell Park. And money talked in those days, even though it doesn't seem like an awful lot of money by comparison. It's just an example of how the dollar has lost its value. Just before I left, Gordon Zubrod arrived and became the clinical director. And he graciously said to me, a senior man, my chief, I don't have a program in acute leukemia. Would you mind if I continued yours? And that was the beginning of interinstitutional collaboration. I went to Roswell Park. He recruited Tom Fry. We became close friends for the rest of his life, which unhappily terminated a few weeks ago. And the outcome was the first paper in combination chemotherapy in the treatment of cancer, which was published in Blood. And Gordon Zubrod persuaded me that, in fact, and Tom, that a single-arm treatment wasn't very useful because you couldn't have anything to compare it against. So we should compare two arms of treatment. And by that time, Abraham Golden, also a scientist at NIH, had shown that these graphs, amethopterin, which is the earlier name for methotrexate, it made a difference how you gave it. Not just daily, as Sidney Farber had done, but if you gave it to mice daily or at higher dose every fourth day, and it depended upon the age of the tumor. On the left, early tumor, on the right, advanced tumor. There were many variations that could lead to a change in the outcome of leukemia therapy. So we put together a study of combination chemotherapy where methotrexate was given daily or where methotrexate was given twice a week. And it's the bottom line shown there. Not very impressive, about 10% survival at 2 years. But you have to look at this in the context that the survival of acute leukemia before this, the median survival was 4 months untreated. And here then the median survival had increased to almost a year. And successively in the collaborative group, and the Cancer Chemotherapy National Service Center was established under the urging of Mrs. Lasker, and Gordon Zubrod suggested that we copy the methodology that had been used in the British Medical Council for the treatment of tuberculosis, a collaborative, multi-institutional set of investigators, and so many cooperative groups were established in the United States. The Cancer and Leukemia Group B is the oldest continuous one of that, but it has since been blended in with other groups and is now known as the Alliance for Clinical Trials in Cancer. And here is a listing by May 1969 of successive protocols, not really related to new drugs, but mostly related to how the drugs were given. And here are the same data in 1994. These are the programs of chemotherapy of childhood acute leukemia during the time I was the chairman of the Cancer Leukemia Group B, and you can see by the time we stopped, 60% of the children were alive without evidence of disease at 10 years. And subsequently, this has become nearly a universally curable disease, with the proposition that getting close to 80% survival now. Well, when I left Roswell Park and went to Mount Sinai, I became interested not only in these kinds of chemotherapy things, but in the proposition of what causes cancer. But before that happened, in 1972, Mrs. Lasker, who's here, and her sister, Alice Fordyce, who's here, flanking Dr. Sidney Farber, who was the first reporter, senior author of the paper demonstrating treatment, temporary remissions in childhood leukemia. And these are the Lasker Prize winners in 1972. There are 16 winners that year. Ordinarily it's 1 or 2, but the Lasker Foundation thought that cancer chemotherapy was so important, we ought to find most of the things. Here is Gordon Zubrod, who was the creator and master of the concept of multi-institutional collaborative groups. This is M.C. Lee and Roy Hertz, who first cured choriocarcinoma with chemotherapy. This is Burkitt of Burkitt's lymphoma and Joseph Birchinall. But essentially everybody else here is either a member or an alumnus of the National Cancer Institute. There were 16 awardees, 10 were members or alumni of the National Cancer Institute, 6 were from other institutions. That's about a good ratio for the NIH. When I got to Mount Sinai, I became interested in why do kids get leukemia and why do other cancers start. And most cancers arise from specific causes. The problem is we just don't know what those specific causes are most of the time. So I was fascinated with the observations made by John Bittner in 1936. In 1936, he recognized that if he took a strain of mice that had been inbred brother-sister matings and had breast cancer as one of its characteristics. Breast cancer here in one of the granddaughters was every individual developed breast cancer. And he foster nursed some of these offspring on milk from mice who were not given to having breast cancer as a familial event. And here, 5 out of 6 did not get breast cancer. And he reported that there was a milk factor that led to carcinoma of the breast in mice. Over the years, that milk factor was identified as the mouse mammary tumor virus. And it is the cause of breast cancer in mice. And even a mouse that doesn't herself have breast cancer as a characteristic can carry the virus and give it to her offspring. But when a mouse whose offspring are foster nursed, the incidence of breast cancer goes down tremendously. And so it was recognized the virus passed in the maternal milk of the mouse, and infected lymphocytes in the upper gastrointestinal tract, and the lymphocytes migrated to the breast and infected breast cells, and they got breast cancer. Sometimes they also got lymphomas. Charlotte Friend was an eminent virologist at Mount Sinai who discovered the Friend leukemia, and when she died, unfortunately, Beatrice Pogo, her principal collaborator, came to work with me as director of the Cancer Center and said she didn't want to work on mice anymore. But I was a clinician, she'd like to work on humans, and I said, if breast cancer in humans is due to a virus, it's going to be a kissing cousin of the mouse mammary tumor virus. And we found a sequence in the envelope gene of the mouse mammary tumor virus, and it's shown in enlargement here from base 975 to 1640 that was absolutely unique. It didn't occur in any other virus, and it didn't occur in the human genome. So finding it, we knew that we would have a footprint of the virus. And we made primers and did the PCR polymerase chain reaction, and the amplicon could be seen in a DNA southern blot. Here's a ladder. These are DNA extracts from the mouse tumors, from the human tumors. This is from a mouse tumor. And you can see that we found evidence of the virus by visual inspection in a breast cancer, but every time we have always done internal hybridization with a radioactive probe, and even though you can't see it in 3, it's also a positive breast cancer. And those are fresh tissues. These are archival tissues with a smaller DNA segment, but again, internally probed, and number 1 is negative, and 2 through 5 are positive, and this is the authentic envelope gene from the mouse, which smears because it was so powerful. So we then extended the PCR reaction out further in the envelope gene, and here is a 2.9 kilobase piece shown in the chromosomes of a human breast cancer. Notice that it's all on one strand, doesn't go to both strands, as is true for MMTV, indiscriminately in several different chromosomes. Well, at that time, again, the Cancer Institute was deeply involved. We got specimens from the Cancer Institute in the Cooperative Breast Cancer Tissue Registry, which included the tumor from a breast and normal tissue from the same breast. And normal tissue from the same breast did not contain MMTV-like sequences. This one was positive in the normal tissue and negative in the tumor, and we think that's a labeling problem. But you see, even if it were true, 1% of the normal tissue and 30% of the tumor tissues, which excludes genetic inheritance. So women acquire this virus. And finally, we increased our polymerase chain reactions till we got the entire virus, which is 9.9 kilobases long, 95% homologous to the mouse mammary tumor virus. All the genes are represented, and only a short area partially homologous to the endogenous retrovirus K10, which is very similar to MMTV. And we think that eons ago, some woman got infected with this, and it's come through human DNA, and we were seeking how do women get infected with this, which is indeed very close to the mouse mammary tumor virus. And because it came from human tissue, we called it the human mammary tumor virus. And here on the top, you can see an authentic mouse mammary tumor virus sequence, and here the several different human mammary tumor viruses. And notice in the long terminal repeat, there are systematic changes in the sequence which may represent either an unusual virus from the mouse that got into the woman, or regression through certain kinds of human resistance to the mouse mammary tumor virus that leads to these kinds of changes in human tissue. And then this is the virus. It looks just like the mouse mammary tumor virus, and this was shown by Daniel Moore 15 or 20 years before us, that there are viruses in human breast cancer that look like mouse tumor viruses. The mouse mammary tumor virus and the human mammary tumor virus are RNA viruses that get reverse transcribed into the DNA, and everything I've shown you is DNA. But now these are the RNA sequences that are coded from that DNA of the human virus, and you can see they're all authentic, showing that the virus picture I just showed you is indeed human mammary tumor virus and not some other virus that's been liberated. And we have insertion sites in half a dozen tumors. This particular one, because the mouse mammary tumor virus and the human mammary tumor virus do not contain oncogenes of their own, they get randomly inserted, and they may impact on nearby genes that are either known to be oncogenes or tumor suppressor genes. These two have some association with cancer in this particular insertion site in chromosome 5. And this turns out to be a significant factor. 40% of American women's breast cancers have the viral sequence in them. If the proband has a sister or a mother or an aunt with breast cancer history, 63% are positive. If breast cancer arises during pregnancy or lactation, it's also more than background, and it is known that there are hormonally responsive elements in the long terminal repeat of both mouse mammary tumor virus and human mammary tumor virus, which may account for the frequency in women as distinct from men. And inflammatory breast cancer, in collaboration with Paul Levine here in Washington, who's an alumnus of the NCI, 70-odd%. Well, when we reported this in our paper, our first paper, and I missed the fact that here, normal breasts in women who've been biopsied, almost always, not really biopsied, but had reduction mammoplasties for breasts that are ungainly or difficult, almost zero. We don't, because they're anonymous specimens, we don't know what happened to these two women, which we would certainly like to know. In patients who have envelope-positive breast cancer but have a biopsy of their opposite breast, 2 were positive. Peripheral blood mononuclear cells in healthy women don't have the viral sequence, but if in fact they have envelope-positive breast cancer, then about 1 in 4, we don't know whether those are circulating breast cancer cells or mononuclear cells, which is part of the life cycle in the mouse. When we published our first paper, Tom Stewart from Ottawa said that answered the problem for him. And I didn't know what his problem was, but his problem was that the breast cancer frequency in cases per 100,000 per year in Eastern Europe had always been much lower than the breast cancer frequency in Western Europe. And he was aware, but I wasn't, of the work of Russell Sage, who had published this in 1993. This is the distribution of mice in Europe and in all the colonies that Europe colonized. East of this line from Denmark are Mus musculus, with very little MMTV in its genome and very low breast cancer frequency. And west of it is Mus domesticus, the common house mouse, which is present in Western Europe and all of its colonies, that has a lot of MMTV in its genome and gets spontaneous breast cancer. And that put an onus on us to go around the world and find out whether or not this was of importance. Stewart pointed out that in the countries that have the house mouse, the common house mouse, 66 cases per 100,000 per year, and they're in the Western world. And in the Eastern world, 21 cases per 100,000 per year. And just to point out this high difference in the lands of Mus domesticus and the lands of Mus musculus. And everybody knew that in Asia, Japan and China, breast cancer was less frequent than in the Western world, and ascribed it to genetics, diet, ethnicity. And we went around the world and looked for the breast cancers and the frequency of the human mammary tumor virus in their breast cancers. And here in the United States, Mexico, Brazil, Argentina, also Australia, another colony of Western Europe, 38 in this particular thing of fresh versus archival breast cancers, but high frequency, with high frequency of breast cancers. And in Japan, 3 positive. Oh, my slide seems to have changed. 12% positive. In Vietnam, 0% positive, 1 out of 120. And in China, 7% positive, and in Iran, 0. So in the Eastern world, less breast cancer and less mammary virus in the breast cancers. In the Western world, a lot of breast cancer and a high frequency of virus. In Africa, and you saw the African had high frequency of the domestic mouse in North and West Africa, we found high levels. And in Tunisia, where we have the most specimens, very high frequency of the human mammary tumor virus. And we were astounded to see that where we had the most African specimens, it was 8%, only 1 out of 12. And then I found Max Parkin's work. Max Parkin is a wonderful epidemiologist who works for the International Agency for Cancer Research. And here in West Africa, red is breast cancer and blue is cancer of the cervix. There are no good cancer registries in Africa, unfortunately, but cancer of the cervix is very common. And except in Tunisia, where culture says that girls should be pale and plump to get married, and therefore they keep them in the house, and there are strict laws against premarital sex. There's very little cervical cancer, but there's high breast cancer frequency in North and West Africa, and compared to cervical cancer, which is about the constant here. It's uncommon in East and South Africa, and that's because they traded with Arabia and China and India, whereas the West traded with North and West Africa. So I presume that there are different species of mice in Africa. Well, we had to show that the virus was infectious, and indeed it is. This is the co-culture activities. Mount Sinai School of Medicine, MSSM cells are breast cancer cells that we know are PCR positive, and there were none in this container, so any positive in the recipient cells would represent laboratory contamination. And here there were no recipient cells, so any positive here through a 4/10 micron filter would represent a filter tear. And we did co-culture experiments using using Mount Sinai breast cancer cells and recipient cells of different kinds. And here are B cells. 3 out of 3 accepted the virus and were infected. T cells, 100%. Peripheral blood mononuclear cells, 75%. And human mammary epithelial cells, which are from normal breasts, commercially acquired, half of them were infected by the virus in co-culture experiments. An infection was defined this way. Here are the recipient cells pre-culture, no evidence of the envelope gene. After the culture, here's the envelope gene, DNA amplicon, and here the protein. It is active in the cell because using monoclonal antibodies, which we've developed in the there's no evidence of the protein, but afterwards, envelope protein appears. And when one takes the cells, these are human mammary epithelial cells to show that there's no vimentin in the pre-culture experiments. And here are Mount Sinai cancer cells showing that they do indeed have vimentin, which is a characteristic of epithelial mesenchymal transition, a characteristic of cancer. Here, Michigan Cancer Foundation 10F cells, pre-culture and after co-culture, they have picked up the evidence of vimentin, suggesting that these Michigan Cancer Foundation putatively normal cells in culture indeed have acquired cancer characteristics by accepting the human mammary tumor virus. Humans get lots of lymphomas, many different kinds. But Dr. Polly Etkind, one of our collaborators, showed that if a woman has an envelope positive human mammary tumor virus breast cancer and gets a lymphoma, which they do at a higher frequency, half of them have human mammary tumor virus in the lymphoma. Exactly like the mouse. The mouse gets lymphomas. Not as frequently as the breast cancer, but they do get lymphomas. Endometrial carcinoma we looked at because it's another tissue highly stimulated by estrogen, and I'll show you that in a moment. And other workers, Mason and Sarkalian, saw our primers. They were looking for the cause of biliary cirrhosis, which nobody knows, so they give it a fling, run the test, and it was positive in biliary cirrhosis also. Endometria without cancer, we looked at 28, none had the virus, but endometrial carcinoma, a quarter of them were positive. This is normal endometrium, and here with immunoperoxidase, well-differentiated endometrial carcinoma, and here undifferentiated endometrial carcinoma, showing envelope protein from the MMTV, the HMTV infection. And how do women get this virus? There is a case report of a technician dealing with mouse breast cancers who had normal antibody titers 3 times in a row and then turned positive. 9 months later, developed breast cancer. A month after that, a mass in her axilla, which was biopsied. And then perhaps direct contact with a mouse. And the mice of this world contaminate our food chain up and down the food chain. I had a wonderful collaboration with the Food and Drug Administration, which inspected the wholesale grocery warehouses where mice and rats frequently visit. They could tell their visitation had been present because by ultraviolet light they could scan for urine contamination. But the FDA then had to change and has begun looking for poisons coming in imported foods and didn't have time to discuss mouse viral activities. Too bad. But we found, as in the mouse, normal women shed human mammary tumor virus in the cells in their milk. 8% of normal women, and there are 4 million births a year in the United States, 2 million female births. 8% of 2 million is enough to account for the 40% of 230 or 240,000 breast cancers every year, if this were the only means of transmission. So we think that this is likely the mechanism by which it gets transmitted. If a woman has had a biopsy of her breast for whatever reason, which really means somebody thought they might have breast cancer, 20% are positive, suggesting that there is a kind of mastitis due to HMTV. The virus has been found by a Russian scientist in Peyer's patches. If a woman or a male offspring, because they also nurse, gets virus into their cells, goes to their breast, the male may not get breast cancer because, in fact, he doesn't have the cofactor of estrogen. But this is a logical mechanism, and we're trying to prove that that is indeed true. We haven't proved that HMTV causes human breast cancer yet, but of course we're following that. Brings me to this parable from the late Frank Schabel, who was a mouse doctor, good friend of mine, who pointed out that humans walk on 2 legs, not 4, drink when they're not thirsty, have a mating season that's 365 days long, but otherwise they're like all the other animals. And I think humans are like all the other animals. I think they're like the mouse in that there's a virus that causes breast cancer in the mouse and that it gets transmitted by the same route, has the same pathological endpoints, and in fact constitutes, I think, evidence that zoonoses are much more common than we recognize, and we all recognize that respiratory viruses from chickens and from swine can infect humans, and we shouldn't be reluctant to think that similar viruses cannot. And to prove that, the next speaker, in his metamorphosis, when he was like a young lion, looked like an animal, but he grew out of it. And so I'm very happy to say that he's here and specializing in infectious diseases. And if I can't prove that the virus causes breast cancer, maybe he and his colleagues can. Thank you. Thank you.
Well, I've known a lot about Steve, but learn something every day. That was terrific. Thank you so much. Steve comes from a remarkable family. Not only is his dad a distinguished clinician and investigator, but so is his mother, Jimmy Holland, who's also here today, and welcome to the NIH again. So now let me tell you about our next speaker, Steve Holland, who received his medical degree from Johns Hopkins in 1983 and continued on as a resident in internal medicine, assistant chief of service, as well as a fellow in infectious diseases. He came to the NIH in 1989 as a National Research Council Fellow in Mal Martin's Laboratory of Molecular Microbiology in NIAID, working on transcriptional regulation of HIV. In 1991, I had the good fortune that he joined my lab, the Laboratory of Host Defenses in NIAID, shifting his research to the host side with the focus on phagocyte defects and their associated infections. His work centered on the pathogenesis and management of chronic granulomatous disease and Job syndrome, as well as other congenital immune defects, including phagocytes, and those predisposing to mycobacterial diseases. In 2004, he became chief of the Laboratory of Clinical Infectious Diseases in NIAID, and in 2011, he was selected to serve as the NIH Deputy Director for Intramural Clinical Research, working very closely with Michael Gottesman. Among Dr. Holland's awards is the 2002 NIH Distinguished Clinical Teacher Award, the highest honor bestowed on an NIH clinical investigator by the NIH Clinical Fellows. In 2011, he was named an NIH Distinguished Investigator and won the Boyle Scientific Achievement Award of the Immune Deficiency Foundation. He received the American College of Physicians Award for Science in 2012, and he's the past president of the International Immunocompromised Host Society and of the Clinical Immunology Society, and a fellow of the American Association for the Advancement of Science. He's also an elected member of the American Society for Clinical Investigation. He's received several NIH Directors Awards for collaborative scientific work, including the discovery of the new microorganism Granulobacter bethesdensis in 2006, and discovery of the cause of Job syndrome in 2008, and the discovery of DOC8 deficiency in 2010. He's an active editor and reviewer and currently serves on the editorial board at the Journal of Clinical Immunology and is a consulting editor at the JCI. And today, he's going to tell us about location, the mycobacterial susceptibility story. Welcome, Steve.
I know there's no pressure here today. So thank you very much, John. It really is an extraordinary pleasure and honor to be here, not only with my dad, what a great, I'm just a lucky guy, but also to have a chance to talk to you about something that I've been working on for a while and to try and explain to you why we've been doing it and to show you where we've been successful and also where we've been less successful. So I have to just give you my disclosures. I really do think she's pretty good. And I'm going to discuss some off-label uses, and you'll have to wait to the end to find out about the interesting ones. So there are objectives that you'll acquire as you go through, so I'm not going to reiterate those now. So we heard about the guy who came here on the first day of patient admission to the NIH Clinical Center. And I want to show you here, these were the last inpatient rounds performed in this building, the Magnuson Clinical Center, in 2005. And you'll recognize some of these characters. This one here, you recognize the tie. This one here, this was before he had the hand transplant. That's really quite attractive, John. And you recognize other people that were involved in the care of patients here at the clinical center. And what I want to do in the next 2.5 hours is walk you through some of that stuff. So now remember, the first Hippocratic aphorism, I'm only going to read it in one language, but he points out that life is short, the art is long, opportunity fleeting, experience misleading, and judgment difficult. And to that I can all say, amen. So when I came from the basic science laboratory to the clinical laboratory, I had the great good fortune to work with an inspired mentor and scientist, John Gallin. And he said, hey, this is just easy. Don't you worry about it. First, you just go figure out how to take care of those patients we already know about who have some pretty strange diseases. And then that's okay. You just go find your own. I said, what do you mean, find your own? But he was absolutely right. It's all about your niche, your insight, your relationship to the clinical disease. And the diseases I want to talk to you about today are those caused by the non-tuberculous mycobacteria. So we know a lot about Mycobacterium tuberculosis, discovered in a kitchen in 1882, but then there are these other mycobacteria, and my lab and others have focused on the non-tuberculous organisms on the premise that if you find people who have infections with those organisms, they must have a problem. They're too common, they're environmental, they're not very virulent, so if somebody's got a bad problem, there must be a reason. And our job is to figure it out. So I'm going to walk you through why location matters, because it turns out, and I didn't get this at first, that it's all about what's the infection, where is it, and that ought to guide you to what the problem is. So there are three real distinct types here: skin, disseminated, and lung. The skin disease is all about contact. And in this elegant set of paper done by Kevin Winthrop, you see here what skin disease looks like in people who have been going to nail salons to get pedicures. And there was this terrific outbreak out in California, and you see here that the number of cases was going up and up and up, and then sharp people like Kevin Winthrop from the CDC got involved and said, gee, let's track this down. They identified a nail salon that was doing pedicures. They closed the salon, and the cases went away. Now, there is no free lunch. And you close down the pedicure salons, there are consequences to this. But the important take-home here, and I want to make sure I give you practical information, this is usually something that happens in healthy people. The inoculation is percutaneous. It's very self-limited, but if you aren't comfortable with that, it can be treated successfully. Only a few species do this, and only certain exposures like pedicure. But the critical element, don't shave before the pedicure. If you take out the shaving, the pedicures really aren't such a big deal. So that's one of those objectives that you got to know. But disseminated disease is really where we know the most. Now, this is the rarest form, but it's also the one about which we know a lot. And these are a big problem. And so my experience with these organisms and these infections began entirely as an accident. I took a phone call when I had just come into John's lab from a doctor in Florida who said, boy, I got this child. He's got disseminated Mycobacterium avium complex infection, and it's involving all these organs. He's so sick he's on parenteral nutrition all the time, and you know, he's got 2 maternal uncles who have the same problem. Do you think this could be chronic granulomatous disease? Well, I'd been in John's lab for a while, and I knew that this wasn't likely to be chronic granulomatous disease. They don't get that infection. But I thought, boy, if there are 3 members in one family who all have disseminated disease, it's likely to be genetic, it's likely to be on the X chromosome because it's being transmitted in a sex-linked way, and that means it must be discoverable and it must be important. So the logical thing to say is, I don't know what this is, but boy, you ought to come to the NIH. And they did. And when they came here, we had just been, John's lab had been studying interferon gamma in chronic granulomatous disease. There was a lot of information about how to use interferon in the treatment of intracellular infections. And so we did it in these patients. And Just to summarize what happened, here's this little boy. He was 5 years old. He hadn't grown in 2 years. With the initiation of interferon, he grew and his nutrition improved. He came off TPN. He was cured of his infection. This man here, who was in his 40s, had 6 liters of ascites being drained from his stomach every week. And with the initiation of interferon, that went away, and he was cured of his mycobacterial disease. Well, you know, that was pretty exciting. And I thought, wow, this is something we ought to really pay attention to. And so I changed my focus and started looking at mycobacterial disease, and we began to try and elicit and understand this pathway in which mycobacteria infect macrophages. They lead to the production of interleukin-12. That works on T cells and NK cells to make interferon gamma. That works on its receptor on all nucleated cells, upregulating tumor necrosis factor, and then driving the killing of mycobacteria through a pathway that is still undefined. And over the years of studying this, we've now been able to put new genes into this pathway and been able to identify new mutations in this pathway that are clearly etiologic in the cause of disseminated non-tuberculous mycobacterial infection. Some of these genes have been identified here. Many of these genes have been identified elsewhere. But we've seen all these patients, and we've tried to use each patient to understand what's the mechanism, what's the pathway, what should we know. In addition to finding things that are genetic and germline, we've been able to identify mutation, the acquisition of autoantibodies to interferon gamma and IL-12, as recently reported by Sarah Brown from our group, clearly identifying that this is the critical pathway that controls disseminated disease. Whether you got it in your germline or you got it later in life, this is the way it happens. And that is true for disseminated disease. So our mantra now is that if disseminated mycobacterial infection, people are just not normal. There's a problem, and if they've got a problem, we can find it. It's usually in the interferon gamma pathway, and it can be genetic or acquired. The exposure is usually environmental, and the disease, if it's disseminated, is usually severe and progressive, and untreated, it is fatal. It's not easy to treat, and they get all kinds of mycobacterial infections. So we're very confident now that it's immune defects that cause these disseminated infections. Now this is where we go back to the first Hippocratic aphorism, that judgment is difficult. Because now if we step over and say, wait a minute, if skin disease is sort of straightforward and disseminated disease is sort of immune, how do we understand the most common set of diseases? And those are diseases of the lung. And these really are troublesome. Not only are they troublesome, they're extremely important. These infections, so here you see curves looking at the rate of non-tuberculous lung disease. This is isolated lung disease. Non-tuberculous lung disease in women and in men over time, 1997 through 2007, and you see that the rate is going up. You also see that the rates are different between whites, blacks, Asians, and Hispanics. That is, there are both age and ethnicity influences here that suggest that this is going to be a complex interaction. In addition, location is critically important, and those states in black have very high rates. You see that these are mostly coastal states, although we're still looking for the coast for this one. These are rates that are very high, as opposed to other states which have rates that are somewhat lower. But to put it in context for you, the non-tuberculous lung diseases, pulmonary non-tuberculous mycobacterial disease, they're thought to be about 140,000 cases in this country. In contrast, keep in mind that the rate of tuberculosis, there were 10,000 cases in 2011, and that rate was down 5% from the year before, and there's no reason to think that rate will not continue to decline. So these are climbing, these are falling, these are 10 times more than this. How much effort do we spend trying to understand this problem? So we thought, boy, let's take what we've learned about immune dysfunction and transfer it now to lung disease. And after years of work, what I can tell you is no. there do not seem to be any significant reproducible immune defects in pulmonary non-tuberculous mycobacterial disease. That is, when we interrogate this pathway, it's just fine. So we thought, well, this is confusing. And we went back and started looking at our patients to ask, are there other things that we missed? If it's not immune-based, what else could it be? Well, there were some interesting things here. So, as had been noticed many years before, we went back to try and confirm, is it true that patients are taller and thinner? And the answer is, yeah. Patients with lung disease, here we had 60 patients with lung disease compared to a very large sample of normals from the NHANES database. And our patients were clearly taller, 165 centimeters versus 161, and much leaner, BMIs of 21 versus BMIs of 28. Now, you could argue that people get thin because they get sick, but you can't argue that people get tall because they get sick, right? There'd be a big market for this in the basketball group. So something must be going on, and it must have long preceded the development of disease, because this is a disease that comes on typically in women in their 60s. In addition to the fact that they were taller and thinner, they had other abnormalities. Scoliosis was found in over half, mitral valve prolapse in almost 10%, and pectus excavatum in almost 10%, suggesting that there were developmental morphologic features important in this condition. In addition, mutations in the cystic fibrosis transmembrane conductance regulator, which are involved in creation of disease cystic fibrosis, these were present usually as heterozygous mutations in a large number, but did not cause cystic fibrosis in these patients, but were present. Well, then working with Ken Olivier and Rhonda Colombo, we were able to look at our group of patients and say, well, wait a minute, if all these things are happening and we think that they begin before disease onset, and we think some of them might be developmental, maybe they're germline. So are there familial associations? That would point us in the direction of genetics as opposed to environment. And sure enough, there were family trees that suggested dominant and some that were consistent with recessive inheritance. So now we've got a funny problem, right? So we've ruled out immune dysfunction, and we've got a disease that is environmental, gender-based, associated with somatic features, looks like it's genetic and likely to be a single gene, at least in some families, chronic and progressive, difficult to treat, and after all that work, what we could say with great certainty was that pulmonary disease is not disseminated disease. Now, that may sound sort of trivial, but that was not obvious when we started. It seemed like they ought to be linked. So in order to understand that better, Ken Olivier and his coworkers went back and said, let's look at the autopsy pathology on those people who have lung disease that have isolated lung disease and disseminated disease with lung involvement. And sure enough, the remarkable thing is that if you had disseminated disease with lung involvement, the inflammatory response was quite poor. Whereas if you had pulmonary disease with pulmonary involvement, the inflammatory disease was quite robust. In fact, when you looked at these are disseminated by nature, right? They are all over the body. But these patients that had pulmonary disease, they never get disease outside the lung. And so now we're stuck saying, wait a minute, it's disease that's limited to the lung and not associated with immune deficiency. How can we understand that? So, as is often the case, you need to look out to a paradigm. Where are the paradigms that we can use to understand what's going on? And the most important paradigm, the most well-studied paradigm, again, that Ken Olivier has really brought to the fore here, is cystic fibrosis. And the really impressive thing is that if you look at the rate of pulmonary non-tuberculous mycobacterial infection in cystic fibrosis, you see that as patients get older, the rate goes up into almost 50%. So this is funny. We've got a disease that has its onset late in life. Cystic fibrosis is a disease with onset late in life. How might we put those two things together? And so now we have to go back and say, wait a minute, lungs are complicated. It's not just air in, air out. There are other things that they do. They actually have to clear out all sorts of debris. And let me just see if I can make this work here. Oh, this is exciting. So, what is it that you want to think about in the lung? It's really that cilia have to beat. And I don't have music to go with this, I'm sorry. Taylor Swift doesn't have a cilia song, though I'm working on it. And what you see is that cilia are very careful, they're very rhythmic, they're very smooth, they know what they're doing to get things where they have to go. And so that got us to think, wait a minute, ciliary function is very important in keeping the lungs clean. And we know that patients with ciliary defects and in cystic fibrosis, although the cilia are normal, all of the secretion is abnormal, and so the ciliary function is diminished. So we said, maybe if cilia are important and people with ciliary defects get into trouble with non-tuberculous mycobacterial infections, maybe we better go look in our patients with non-tuberculous mycobacterial infections to see if they've got ciliary defects. And so Cedar Fowler, a student in the Oxford-Cambridge program, came in and helped us really begin to study how might this work. So the truth is that, although the lungs are down here and sort of difficult to get at, there are easy ways to get at the same sort of cells, and those are on the inferior nasal turbinate. If you don't think that your turbinates have that, just come by the lab and we can help you out by proving it under the microscope. And what you can do is scrape that inferior turbinate, take those cells out and culture them, or you can take them out and look at them under the microscope and look at their ciliary function to show that they are normal. And in fact, you can count the beat of the cilia and develop something called the CBF, or the ciliary beat frequency, which is important in how you clear things. And sure enough, when we started to look at our patients with pulmonary non-tuberculous mycobacterial infection versus other groups, what you see here, normals have a CBF in our hands of about 10, but the pulmonary NTM patients had a CBF of 8. Patients that have intrinsic and severe defects in the cilia, known as primary ciliary dyskinesia, have rates that are much lower, around 5 to 6. So that got us very interested in how might that be important. Is this an acquired problem? That is, do they lose ciliary beat frequency with age? And so we went back to look at what happens to CBF over age. You can see here, this is the CBF of ERDAs, and you can see CBFs of postdocs and then CBFs of senior staff. But you also then see that the rate of ciliary beat frequency in these patients with pulmonary non-tuberculous disease is relatively stable over a long spectrum of life, suggesting that this is not just an age-related phenomenon. And in fact, it's not caused by infection, which would be another thing that you'd think about, because when we add mycobacteria to cultures of these cells, in fact, their ciliary beat frequency goes up, doesn't go down. And if we take conditioned media from these patients and we put it onto normal cells, it does not alter the ciliary beat frequency. So we don't think that this is a transmissible inhibitory effect. In fact, we think it's intrinsic. And so finally, to understand that, we were studying how these cells respond to external stimuli. And for a variety of reasons that have to do with our studies of nasal nitric oxide production, we used a nitric oxide donor, S-nitroso-N-acetyl-penicillamine, or SNAP. And when you put SNAP onto things, it liberates nitric oxide that then can be used by the cell to perform its activity. And sure enough, when we take our patients with pulmonary non-tuberculous disease and add nitric oxide to them, their ciliary beat frequency normalizes. Well, SNAP is not an approved medication for the treatment of humans. However, sildenafil is. And that also inhibits phosphodiesterase 5, leads to the increase in the levels of intracellular nitric oxide, and sure enough, sildenafil increases the beat frequency in patients with pulmonary non-tuberculous disease right up to the normal level. Now that's all fine in vitro, and maybe, if our hypothesis is correct, that ciliary dysfunction in the lung is involved in susceptibility to pulmonary mycobacterial disease, and we can upregulate ciliary beat frequency in vitro with Viagra, what can we do then in humans, in vivo? And this is preliminary data, and I'm showing it in this sheltered environment, because I know there's a fence around the NIH and we're safe here. But these are preliminary data, but provocative preliminary data. And here you see what happens after the first dose of sildenafil. This is 30 minutes after the first dose, so pre and post in patients who have pulmonary non-tuberculous mycobacterial disease. This is at day 0. They're still responding at day 7 and still responding at day 30. Now, whether this translates into clinical improvement, that's what clinical trials are for. That's what the next phase is all about. But so far, we've got a logical hypothesis, we've got compatible findings, we've got in vivo responses that are compelling enough to make us believe that we should be going forward to learn more about this. So, pulmonary infections, it's a respiratory disease. It looks like it's genetic, and we've now got data that we are mining to try and find out what these genes are. Remember that in the case of primary ciliary dyskinesia, there are dozens of genes involved in PCD, and so it may well be the case that there are going to be multiple genes involved in this. It's not going to be a solo problem because there are obviously complex features to this disease, such as height and morphotype. But it really is not an immune defect, which makes sense because this disease never disseminates. There are intrinsic ciliary abnormalities that can be altered. That is, they are druggable. So the take-home message after now over 20 years of studying this that I have finally come to is that, in fact, lung disease is lung disease, and pulmonary non-tuberculous mycobacterial disease is an airway disease, which helps to put it in the context of all those other airway diseases that now lets us understand how would we go about trying to alter airway function and airway defense. So I've tried to give you a sense of how location really matters in this. Skin, disseminated, lung. And it turns out that where your problem is has a lot to do with what your problem is. And so the first question to ask is not what's the organism involved, but where is the organism involved? And try to go back to what the patient has and the patient experience, and not try to cram it, as I did for the first 20 years I studied this, into the model that you understand the best. So I want to finish by talking about that other kind of PCR, patient-centered research. It starts with patients. It's complicated and confusing. It's got lots of twists and turns, but my God, it is the most exciting thing out there. And it really is what the clinical center is all about. And if you didn't know that for sure, here you have the guy here was on day 1 talking about patient-centered research, and here we are today doing the same damn thing. That is really what 60 years of this seems to be about. And I think we've got every reason to be excited about the next 60 years. I'll just acknowledge the people that have done all the work I have shown, and I'm just the vehicle. It's an extraordinary team. It's an extraordinary place. The opportunity to collaborate, to work with smart people who do these things is just amazing. In particular, in the pulmonary disease, I want to acknowledge Cedar Fowler, Ken Olivier, and Doris Wu. And in the autoantibody and the susceptibility data, Sarah Brown, Elizabeth Sampaio, Sergio Rosenzweig, and Krista Zerbe. And in our other work that's been leading all up to this over many years, Alexandra Freeman, Gil Wiesel, and our extraordinary team of study coordinators, nurses in the clinic, and our coordinators who have done so much to get us through this. Finally, nobody does these things without a mentor. Nobody does these things without a family. And for me, it is my enormous gratitude to thank not only my mentor, John Gallin, but my parents who helped to show me why this was exciting and how to get it done. Thank you very much.
So I just want to thank both our speakers for what I feel was an incredible start to a series of grand rounds saluting the 60 years of patient-centered research here, and invite all of you to join us for a reception just beyond these walls behind us in the South Lobby. So we'll have informal discussion now.